right, it's going.
I was gonna wait to start recording until I could at least say, hey, we're gonna start recording.
Sure, I will pause it.
Go. There we go. So yes, just keep that in mind. But then we will be, of course, posting the recordings. CARC has a YouTube channel for all their recordings. So we will be putting them there going forward. But today's talk is about what data to preserve. Like we've all been talking about how there's tons of microscopes coming onto campuses that give off just enormous quantities of data, and it's just not sustainable to keep all of those files. And so you have to come up with a way to curate your data and decide what should you actually keep. And then to introduce our speakers today, I will hand it over to Galen.
Hey, so today we're joined by Huajin Wang, who's a librarian and program director for open science and data collaborations at Carnegie Mellon University Libraries. And we also have Susan Ivey, who's a research data and infrastructure librarian at North Carolina State University's libraries. And so, ladies, if you wouldn't mind, go ahead and give our participants a little background on what you do, a little bit of what you do on a daily basis, and then we could roll right into the presentation.
All right, sounds good. Thanks, Galen and Debbie, for inviting us. I'm currently a librarian at Carnegie Mellon University. So a lot of work I do is actually related to data sharing, open science, and I'm an advocate for open data sharing and for the reproducibility and reuse of scientific data. And I'll also facilitate the collaborations between researchers around research data, how to best use the research data. And before I joined the libraries, I was actually a cell biologist myself. I did my PhD in Canada and a postdoc at Yale and Harvard. So I did have a lot of experience and struggled a lot with the confocal microscopy data. So Susan?
Yeah, hi, so I'm Susan Ivey, research data infrastructure librarian. I work both within the libraries at NC State and with our central OIT office. So within the libraries, I help support our current data services and help develop new services. With OIT, I help with data storage and transfer services. And most recently, most of my focus has really been on identifying services that exist around campus that have to do with data support and computing, and working with some groups to help identify ways we might better highlight those services and get the service providers communicating a little more and just making it a little smoother for our researchers. My background is I am a librarian, a data librarian. I curated research data at my previous position at Duke University, and so I really come at this topic from that aspect. I am not a researcher, and I do not produce or do research with this kind of data. So. All right, you want to get started?
All right. Yeah, all right, thanks. So yeah, I guess like with a diverse background, we can approach the question from a different angle, right? So I'm first gonna start wearing my researcher's hat and share a bit of my experience working with confocal data. Oh, yeah, okay. So for those who are not familiar with the confocal microscopy, so confocal microscope is a microscope that's equipped with a laser, several lines of laser that can has a very narrow bandwidth that can focus on the very narrow area of the specimen or the sample. So in fact, you can not only the area XY wise, but also like the depth of the specimen, you can focus on the various defined depths into the specimen. And at the same time, the samples are usually labeled with specific dyes of different color, and that's targeted with to a specific protein or DNA or whatever you want to label it. So the idea is that protein A is labeled with green, protein B is labeled with red, and then they can only be each excited by specific line of laser. So with this combination, you can only see one specific project, the object at the desired region at a time. And sometimes with the if you want to step up and down in the different depths of the sample, you just have to adjust the laser focus, right? And then often the experiment also requires you follow the sample for period of time so that you come up with the time series data. So this is an example of the data that I generated when I was a scientist. So that was, so this panel is an example of what you would see in the publications. This is published data, right? So I have a green channel here, these dots, and I have a red channel. Even though they're labeled with dyes of different color, you see them initially as a black and white images. And then you would overlay these channels to give you a composite image so that you start to have an understanding what's the relationship between the green objects and red objects. In this case, the green dots are almost always adjacent to the red tubules, right? So this gives you an overall understanding of what your sample might look like. But this is a very simple image that's used in the publication. And with a little bit of supplemental data, you can also include more of the time series, right? With a movie, now you have more information. But still, this is it in the published format. But what does the raw data look like? So this is the raw data. I have, for each channel, I have time series that containing 60 time points, and I have the Z-stack. This is the depth that I mentioned before. The 5 slices for each time point. Then basically you end up with 120 slice images for each channel, and which is extracted into a single image in the published format, right? And then compared to the more polished data, you can see that these images also look more pixelated, more noisy, and less smooth. And the region of is also larger, right? And in terms of data format, because these data are captured with a Nikon microscope, so the format is ND2 files, which is proprietary to Nikon. So in order to transform this into a published format, so this is what I do. First, I need to open this proprietary file format with the Fiji, which is open source software. And then I save that as a tagged TIFF files so that I can do the further processing. And then further, I do some basic image adjustment, brightness, contrast, and levels. And I do the adjustment in a way that is across the board to all these images so that they're comparable with each other. And then now the next step, I want to pick of all these 100-something images, I want to pick 1 or 2 that I want to present in the paper. So what I did was basically pick the middle slice, which I can see most of the activity, and pick at the time point that's kind of support what I want to say. Then from here, I would just crop further the area of interest, and then if needed, in this particular image, I didn't do any deconvolution, but often researchers would also do some deconvolution with their images using third-party software or using their homemade algorithm, just basically to make the images look nicer and more persuasive. But often the problems is quite debatable whether deconvolution is the best practice, because you could very easily introduce artifacts and distortion, so making things less interpretable. So in this case, I didn't use the deconvolution. So basically what I did was just to overlay the green and red channels and added my annotation, like the regions of interest and scale bar, and saved everything as a TIFF. So this goes into the published format. As you can see from the process, this is not a particularly large image set. This is, I think, more standard nowadays for confocal images. But majority information is actually not kept. Also, a lot of intermediate processing information is not there. So the question is really, how do I want to share the rest of the data, what's not in the paper, to make it follow the FAIR principles, right? Findable, accessible, interruptible, and reusable by others. So its easy way might be, you might think it's easy to share these just raw data in the public repositories, right? There are a lot of generalist public repositories like Figshare, Dryad, Dataverse. The problem with these repositories are their metadata are very general. This is one thing. So they are not specialized to capture all the details in the confocal microscopy data, which might include the metadata for experiments, metadata for imaging, and also metadata for analysis, right? And then on the other hand, there are also these specialized data repositories for cell imaging. I'll come back to this one in the end, but so these are way better than generalized repositories, but they're also not ideal. So now I want to walk you through some of the challenges of sharing the confocal data. So the aim for sharing, like I mentioned before, I want to make the data fair. So the question to think is basically what is needed for others to be able to understand your data, to be able to scrutinize or reproduce or reuse the data sets you generated, right? So for the data, for the raw data, I want to be able to share all the relevant slices and time points. And not only that, because often to come up with this one set of experiments, one conclusion, I might want to repeat my experiments 3 times before I'm comfortable of publishing it, right? And then with all these confocal data, there's complex metadata. I mentioned a little bit before. So in addition to the general file names and etc., you also want to tell people how you organized the stacks of the images. Which one is the time point and the z-stack, right? And you want to describe how the examples are prepared, what are the experiment conditions, what microscope setting or the camera setting were used, whether the experiments were done at 37 degrees or at 25 degrees if it's live cell imaging. And also like the whole lot of afterwards image processing analysis methods that ideally would be documented, right? So this is just very complex for the general repositories to manage. And I wish I had more answer here to say what schema to use and all that questions, but this is really a big discussion among the community to kind of come up with a consensus. So with the general repository, you can deposit the data with some general metadata, but now the README file is really important to capture all these other information. But whether this is good enough, I think not. So this is on the metadata side. And also on the data side, it's really not that easy to decide what data are actually relevant and to keep all these replicates from different experiments. So taking the data set that I just shared, for example, so I think many of you know the pain point is like the data set is really large, right? So for the data set I just shared, one image is 1 megabytes, and I have like 120 images for one data set per take. And then that has time to, if I want to share all the raw data, I want to multiply by the fields per sample that I took, and then the number of experiments, and also I have multiple figures in one publication that support my argument. So you can easily come up with like more than 100 gigabytes of raw data, and that does not even include the experiments that's leading up to your final experiment design, and also the data analysis or intermediate data that's produced by data analysis steps. And for most of the, so for data repository at CMU, we use GitHub, which is based on Figshare. And we provide 1 terabyte of free storage space for each researcher. So you can see this moderate data set itself already would occupy a big chunk of the storage space, right? And as far as I know, a lot of institutions are not able to produce even this much. Okay, so this is a moderate data set. And another project I was involved in was the genome-wide screen data. So this, so I think nowadays this type of experiments are more and more common now. You want to look at the phenotype for every gene in the genome. So when we're talking about the human genome, we're talking about close to 20,000 genes. And then so multiply which for all the images we have to take for each gene. So basically we easily end up over 1 million images altogether. So this data set that I had was 1.2 terabytes. So this is a big headache for storing in the data repository, in the general data repository, right? But this is just the beginning. And from raw data, in order to extract useful information, we also use the third-party software to analyze the data to like make image segmentation and to extract, identify which are the interesting objects in the fields, right? So here you can see, like I can see by my eye, but I cannot quantify these objects unless I identify which other objects of interest. So this is what the data segmentation step tells me. And so all these gives me the first set of intermediate data. And then after this, I extract all the information, the numbers from the segmented images and export everything into a spreadsheet, multiple spreadsheets, which turn out to be several gigabytes. So this is a second intermediate data set, which is very, very large for spreadsheets. And then all the later downstream analysis to statistical analysis and machine learning models to identify the useful genes are actually based on this spreadsheet. So now I want to just pause here and maybe let you think for a moment of this data set. What would you preserve it? I don't think I will have answer for you, but in the end, I will come back to that. I will let Susan tell you about some of our thoughts.
Great, thanks.
We did have a question for Wajan. How is GitHub funded at CMU, and who is responsible for training users and using it?
So at CMU, we have a partnership with Figshare. So the libraries basically pays for a contract with Figshare. And then we are also sort of development partners with digital science. So I think they allow us to have a little bit more of the free storage space in to have our feedback. So what was the second part? Who are the users?
Yes.
So the users are
for training users.
Sorry, the training users? Oh, who are responsible for training the users? That's a very good question. I don't think we have a systematic way. So usually a lot of like the people come to us to ask us about like how I want to deposit data. A lot of this, partially this is driven by the funder and publisher mandates, and partially is like the work of the liaison librarians. For example, I'm a liaison for biological sciences, and then we tell them about all these, and then they just try it out. And for training, usually we just, if their data is complex, we require that they come to meet with us for a short consultation session before we proceed.
Yeah, and I'll just add on for NC State, we also do 1 terabyte of storage for data per researcher. And in fact, we've increased that slightly, though we haven't widely announced that. We're still working out policies. But everybody will get at least 1 terabyte. And then for projects up to 10 terabytes, generally funded projects, we originally. And then through the libraries, we offer Dryad. So we have a Dryad membership, and that's our data repository for sharing. And the libraries is responsible for the training. So it's similar, but a little different. So, any other questions before we move on? All right. So I'm going to switch gears a little bit and talk about a resource that a group of us created in order to sort of deal with this kind of data, particularly with the audience of data curators in mind, perhaps data curators that are curating data for deposit into a repository, or those that are just helping with general best practices for data management. So in 2019, the Data Curation Network, which is the DCN, held a curators workshop at Johns Hopkins University. And they have had to date, I believe, 3 of these workshops. And the focus of these workshops is to increase understanding of data curation practices, for people to share their expertise, and just provide an overall networking opportunity for all of those in attendance. The deliverables of those workshops are data curation primers, which are created by 1 or more of the attendees, and they detail curation steps for a particular type of data. And these documents are peer-reviewed and made openly available. And the primary audience, like I said, for these primers are generally curators that have a need to curate data sets for deposit into their institutional repository or another repository, or that just help overall with data curation for reproducibility or for just data management best practices. So our group formed in 2019 to create a primer about confocal microscopy data. What we did was we kind of met to learn more about the data type, to recognize all of the challenges that we just talked about that are associated with it, and to create a template for best practices as a primer. So the members of our group include both of us, as well as Amy Koschoffer and Gretchen Sneff, and Lisa Johnson from the Data Curation Network was our team mentor. Next slide, please. So in order just to give you a high-level overview of our primer, we've included our table of contents on this slide. So you can kind of understand all of the sections and things we talk about. But for the purposes of today's discussion, I'm going to focus mainly on what's in red, which is the preservation action section. Within this section, we created some guidelines for data and documentation that should be kept and shared when sharing data or curating the data. And within this section, we give 3 different scenarios for what data and documentation would be included in a data deposit into a repository. These include the most reproducible and reusable practice, what we consider a good enough practice, and what we consider probably the minimum practice. So this slide you're looking at right now is what we decided would be most reproducible and reusable. So we suggest depositing or keeping process data, which would include the transformed data files, which are converted to TIFF, the analyzed files, and the annotated files. And we also note that researchers should preserve those proprietary raw files and other data as resource and storage allows, and consistent with backup, best practices for backup. And if a funder or publisher requires that all of this data be accessible, which sometimes they may, if you can link from a repository submission to the location of the raw data, that would be ideal. We also suggest a README file, which would include detailed sample descriptions, as well as detailed protocol to create the process data. And lastly, descriptive and technical metadata. Next slide. This is what we came up with as a good enough practice. We think this would probably fulfill most publishers' mandates for publication. And within this, we suggest preserving only the transformed data or what was submitted to the publication, and a README file, which would have the sample description, the protocol to create that process data, and descriptive and technical metadata. Next slide, please. And lastly, this is what we consider a minimum practice. That would be preserving just what you receive, even if it's just the raw and proprietary, or if it's to transform, the processed or analyzed, or the annotated. And we do note in here, if you receive a compressed file type, there is no action that can recover those lost bits. And a README file, which would have the minimum sample description, the minimum protocol, and descriptive and technical metadata. And so both for the good enough practice and the minimum practice, we do suggest always requesting more information and documentation in order to receive the most reproducible and reusable data, or data that you can make into reproducible and reusable data. Next slide, please. Because overall, the goal is always to curate and preserve the most reproducible and reusable data in order to make it as fair as possible. But there's always a balance there. Anybody that's worked in this role knows that oftentimes depositors might be under a time constraint. They might not be interested, and you can do your best to talk about open and why it's great to share and reproducibility. But it's always a balancing act. Next slide, please. So I also want to include something that we consider an important component in our primer, and something that we've gotten a lot of really great feedback on, which was the curation workflow decision tree. So this flowchart contains a workflow for confocal image specific curation questions. We'd like to note that generic data curation questions should also be inspected, but they're not included in this chart. So we see this mainly for this specific data type. Next slide, please. And we do acknowledge within our primer, and we'll acknowledge here as well, that we did have some unresolved issues and questions at the time of publication of our primer. We were under a time constraint, and perhaps we don't have the expertise to answer these questions, but I wanted to share a few of those that are listed in our primer. There are a couple others than what's listed on the screen, but these here have an impact on the amount of data to preserve and share. So in terms of the Z-stack, we do recognize that researchers may want to consider when it would be appropriate to archive or share an entire Z-stack, even if that whole stack is beyond the scope of their current analysis. Future users of the data may benefit from having the whole stack in ways that cannot be anticipated at the time. And it's always a balance though, because Z-stacks can add up to quite a large collection of files. In terms of handling the Z-stacks, we had several lingering questions, such as, do the successive images of a three-dimensional stack of images follow a file naming strategy that identifies the sequence effectively? How does the target repository handle file structures, if at all? Should a Z-stack be zipped or compiled together in its own folder? Did the researcher create a GIF showing the image to an in-depth of field? And if so, should that be preserved too? And then lastly, similar to Z-stacks, we recognize additional considerations should be given in terms of composite images. We know that care should be taken to weigh the cost and benefits of saving single channel images and or composite images. And on one previous slide, we shared the cell image library, and that is a well-curated cell imaging database. And we looked at that a lot when creating this primer. And the cell image library includes a detailed viewer that highlights the advantage of retaining single channel data, and because you can turn different colors on and off, but that really comes at a cost of storage and curation. And obviously that's not something that a lot of repositories might have. Next slide, please. And then lastly, we just wanted to note that there are other DCN primers out there. They're all shared within a GitHub. We've shared a link to that GitHub within the notes document. And we also have linked directly to our primer that we're discussing today. All right. Oh, and lastly, we did want to mention, and we want this probably to be part of our discussion, hear your thoughts. We do think that our primer, though it's targeted at confocal images, could be relevant and applicable to other data types that share similar characteristics, particularly in complexity or size, or the level of processing and analyzing that is required. So we do see some benefit to this, perhaps this primer for other data types, and we'd be interested to get your thoughts on that. I mentioned that when I was at Duke, I curated data, and we worked with multispectral imaging data sets, and I think some of this could be relevant for those, and I know there are probably a ton of other data types. All right, I'm gonna pass it back to you now.
Thank you. All right, cool. All right, I guess we can the primer was just kind of getting things started, inviting the discussion. And you can see we sort of have brought up more questions than answers. So the next, I want to just share a few use cases, and I would like to hear what you think we should do to preserve this data, right? So the first use case is imaging data set. This is sort of comparable with the first data set that I shared from my own experiment. So this is moderate image data set with a Z-stack and time series that's related to a publication. Something unique about this data set is that the researcher himself, he's a computational biologist. He has a specialized software that was published previously, and the software was essential to create the computational model to extract information from this imaging data set. So his intention initially when he approached us was to, he thinks the intermediate data was very valuable and took hundreds of hours of computing time to get there. So what he would like in the ideal world, he would want to preserve the raw data, intermediate data, and also at the same time host the computational model interactively in the repository so that people can run the data and play with the data. You know, that's the ideal world. So what do we think? So I guess you can enter, like type in chat or unmute yourself. I'm not sure with the small, like with the Zoom, how interactive we can be, but so I guess it was, to many of you, it's probably clear at the last point he wanted the interactive computational parts is not going to happen. There are platforms that can do that, maybe, for example, CodeOcean, but they don't give you a lot of storage space. Any thoughts?
Why not hold on to the raw data, the original raw data, because it seems like you could always go back to that, and that seems like the case for a lot of different disciplines. Why is the raw data not the primary target for preserving?
I think that's a very good point. Like often the raw data is the most valuable part. And then for this specific result, I think the intermediate data was deemed valuable simply because to save somebody hundreds of hours of computing time and also to setting up the software and computing environment. So yeah, but I can share what we did actually. Ended up we did like exactly like you said. So we decided to share the only raw data in the GitHub in our institutional repository, and then the rest of it, the PI just decided to share on his own website because the software was already there, and then people are used to downloading data from his website. I have to give him a lot of credit because he has very well curated website, so this helped. But then if you think about it, so the raw data shared on GitHub help this data set to be more discoverable, and then from there you can link to the intermediate and for the downstream interactions.
Another thought I had in this space is with the value associated with the intermediate data because of the compute times. This is something that reminds me of sequencing genetic data in days of yore, that say preserving that for maybe a set period of time, maybe like 5 years, and in 5 years that compute time is no longer as valuable because it doesn't take nearly that amount of time to so to set time limits around preservation, or time limits for when it should be reviewed to deem what's still valuable. And going back to, I'm sorry I didn't catch the name, it's the G, maybe Galen, but the raw data is the most reusable, and it is the most reproducible often because of issues like that.
Yeah, I think that's a great point. Actually, I didn't think about that before. The computing time actually decreases over time.
Yep, and I'll
raw
piggyback on
data is
that.
still.
As somebody who has more of a focus on at a research computing center, there might be an opportunity with a future project, even though this is completed, to point that faculty member to a computing center where hundreds of hours might be readily automated or made easy enough that that work could be completed more cheaply than, say, storing the data for a long amount of time, especially if it's large.
So you're suggesting so the computing center can help faculty to automate the computing pipeline, and then they wouldn't
Right.
need to save the intermediate data anymore?
Exactly. Yeah, at some point, you know, working to make the computing more streamlined can mean that it really is easy enough just to redo that step and reproduce the intermediate data rather than needing to store it. And we run into this quite a bit with imaging data sets or genomics data sets, for example.
Okay, that's also a great point. Okay, thank you. Sounds good. So the second use case is actually not an imaging data set, but like you said, it's a genomics data set that share the similar problems. So the researchers who work on this project, they have been generating the bulk genomics data for a database for echinoderms. So at the time they approached us, basically the person who's working on this project in the lab, you know, was leaving, and he was wondering what to do with specifically the terabytes of intermediate data, because the raw data is somewhere, and then the terminal data was in this like Echino base. But in intermediate data, his thought was that probably nobody else in the lab would use this, and probably it's very unlikely somebody would need it, but in case they would, then it would save somebody lots of time. So that was the thinking behind it. I guess this is a similar problem, but I wanted to hear again, like what are your thoughts, how to preserve, or what data to preserve and share? And what would you do if you want to make a recommendation to the researcher?
It's probably more important to preserve the methods of processing the data, the software necessary to produce the intermediate data, than it is to save the data itself. Just as you said, going back to the previous conversation, that processing time is something that should decrease as long as all of the materials are available to reproduce that processing. So definitely the raw data and processing details should be preserved, and any associated processing software or code.
Yeah, I think that's a great point, that as long as the method of processing is preserved and importantly can be found and linked somewhere, the computing time, I guess, would keep up. So in the end, what we did is just to recommend, okay, so since the value sounds like the value of the intermediate data is not less likely to be great, and considering the size of the data, and we recommended just to basically keep the intermediate data somewhere in the glacier or something, but create a metadata only record in the KillTop so that you have a DOI, so people can find that the intermediate data exists, and then they can request it directly if needed. I think this may not be the best solution, but then considering the resources.
We had a couple questions come up in chat. First, back to the previous use case, the data on the PI's website, is it a direct link or is it request-based? And where is that data stored?
So yeah, on the PI's website, there was a click-through link. So you can just click on the zip files and download. I think that they might live in the cloud on campus in the School of Computer Science computing cluster, data server.
And then another question was, does the computational facility have access to a hierarchical storage management system. Maybe the best place for the computed data is on tape or something like Amazon Glacier.
So at CMU, we don't really have a centralized storage server or space for data. I think most PIs, a lot of PIs would use, okay, biology, there is a server room that every PI has set up a server in that little room. And then the School of Computer Science have their own storage server. And then some would just use Amazon Glacier or S3. So it's very decentralized. I think campus computing is working on this. More questions? Okay. So finally, I want to just come back to the last use case, which was the genome-wide screen database that that was part of my project, but I don't have to worry about this anymore. Just to remind you, the problem is like the raw data was too large to be deposited, but we consider it very valuable because people can easily just take the raw data and just redo all the analysis. And I mean, not easily, but people can take the raw data and redo all the analysis and make new discoveries. And then the first set of intermediate data was also very complex. The image segmentation, very complex, computationally expensive. We did have to use the HPC cluster to do the calculation, months of calculation. So now we're not only talking about hundreds of hours, right? So this is a different scale. And even transferring data up and down from the server was a huge headache. And then afterwards, the second set of intermediate data is these spreadsheets. They look more manageable, and most of the downstream analysis was actually extracted from the spreadsheet. But then you have to trust, basically, the upstream analysis segmentation was good. So any thoughts on this? So oh, sorry.
No, not at all. This might be another good candidate for that idea around putting a time window around how long different data sets are kept, where you find that intermediate number one, the image segmentation is computationally expensive, but no one's using it. So, if you have several years of evidence that intermediate data number two is the stuff that people are really tend to reuse, and intermediate number one isn't being reused. Finding a cheaper or a less expensive storage space for that is perhaps a good decision to make. And with the first challenge of the raw data being quite large, so I know there are a couple of the generalist repositories, at least, that are making provisions for connecting some of the larger cloud storage like AWS to the repository itself. So trying to facilitate some of those large storage issues and associated download issues and things like that. So it's a difficult challenge right now, but maybe in 6 months it'll be less difficult challenge.
Right. Well, I guess time will tell. So I think for me, this data, I think is obvious thing is the raw data need to be kept, but where is still not like, I guess I'll share more in the next slide, but it's not for many places, the where is still a big issue as of now. But I want to also mention the intermediate data, the first set of intermediate data. So many people wouldn't use it directly, but if I were to look at a screen data, like if I were to decide whether I want to use the spreadsheet data generated by somebody else, I would probably want to go back and look at their segmentation and look at how the quality of the analysis before I can trust this output, right? So in my opinion, this is still somewhat relevant for people in order for people to reproduce and reuse the data. I guess all this is to say, I wish I have better solutions. But if I were to do this today, I would probably keep the raw data and maybe share some examples of the segmentation just to demonstrate whether, like a sample data set to show that the algorithm works and the segmentation was good, and then to share the spreadsheet data so that people can use it. So from here, actually, this is something I recently found. I was very excited about this. This is an image data resource. They put this together in 2007. So I think it started from researchers in Europe, like Ambo was probably spearheading this. So they specifically they don't care about how big your data set is. They want complete imaging data set that captures not only one set of experiments, but the whole study. And they have the infrastructure that can integrate different experiments. They have the standardized and metadata that's integrating what I mentioned before, like experimental imaging and also like analytical metadata. And also these metadata are mapped to common ontology, and the gene names and phenotypes are also mapped to other databases like protein database or the genome database. And also there's some amount of data visualization and analysis pipeline built in there, so you can actually sort of play with the data. So this is kind of ideal world for me to preserve this type of data. But now, as far as I know, so far they are quite selective of what data to accept there. So I guess at the end of the day, if I come across a data set like this, I could recommend researchers to go to this resource, but then there's no guarantee they're gonna take it. So this is the end of my slides, I think. And I really enjoyed the process of working on this primer with all these co-authors, Susan, Amy, Gretchen. So we kind of, like Susan mentioned before, we met each other at this data curation network. For myself, I didn't know much about specialized data curation. And then we learned a lot from this, like working with this data, taking the workshop and working together as a group. And Lisa Johnston, our primer mentor, was also a huge help. And here's how to cite us if you want to use or adapt. So this is it. Do we have questions?
We did have some more in the Google Doc. So a question for Susan. Why do you recommend TIFF formats instead of something else?
Well, I wouldn't say that anything else would be terrible, but I think we landed on TIFF because it's it can be a lossless format. Yeah, somebody just chatted that because it's lossless. That's why we recommend TIFF. And I know that it's still a large size. I don't know if we have several authors from the primer on here if they want to add anything to that, but.
Yep, and we posted a list of the current primers in the doc. And there was also a question about how do we request a new primer?
Yeah, I saw that question, and I don't speak for the DCN at all, just a workshop attendee. But, I do know that if they continue these workshops, which I don't know if that's their plan or not, they would probably be interested in at least knowing if somebody had interest in specific data types, because I do know that at our workshop, there were some people that weren't quite sure what they wanted to work on. And so if they had a list of suggestions, perhaps somebody might be interested that attended one of those. But again, that would be a question for them, but I'd be happy to pass it along.
And then expanding out from confocal microscopy, what other disciplines have begun taking advantage of these data management services at your campuses?
Was the question has been, or?
Yes, so at your campuses, is anyone besides confocal microscopists coming to you for data management?
Well, at NC State, we serve all of our disciplines, so I would have to pull our stats, but we get a wide variety of questions. We're, you know, STEM school, we have a big ag focus. So most recently in memory that I can think of, it was a lot of agricultural from the College of Ag coming to us, generally with data management planning questions, and then trying to identify data repositories to fulfill their funder mandates. Aside from that, I get a lot of questions about how to store and transfer big data. And that comes from all over as well. And that, you know, that's because I also work with the office OIT. And so that's sort of where we stand at NC State.
So at Carnegie Mellon, we have a lot of requests from social sciences, and also, oh yeah, one big domain is the psychology with the neuroimaging data.
Does proprietary versus open source file type factor into keeping it or not, do you think?
Yeah, I think it can. And we went back to a lot of the times the raw is a proprietary format because of the machine that it's coming off of, which is why the first step I think generally is to convert it to TIFF, so to work from the TIFF, which is probably why, I don't know, we might, I think this is an opinion, but you might say to keep the TIFF, but the proprietary raw, if you have a way to preserve that for perhaps future use, that would be fantastic. I don't know, but that's just my opinion.
Yeah, I think like a lot of the proprietary data format can be opened by this Fiji software. So actually this is one of the consortium OME, like open formats that they're working on. But still, I feel like it's not the best practice to keep the proprietary.
For the use cases that you presented, was there actually funding to keep everything for them?
We promised 10 years.
And question in chat, is there data loss, image or metadata converting ND2 to TIFF?
I think it's lossless. That's my understanding. Oh wait, like, yeah, right. So if you convert to TIFF, like then if you convert to tagged TIFF, then the metadata should be saved in the meta information.
Any more questions?
Yeah, there was one more. Could either of you share perhaps a success story of where preserved data found productive use in a new or different project? And you know, in your experience, is that a rare thing, or is that something that does happen quite often, and perhaps often enough to guide the methods that are used in preserving data? That's a big question.
That's a very good point. We have to find those cases.
Yeah, I mean, like, you know, when have those cases happened? And you know, it seems kind of random.
Well, I guess I've definitely seen more reuse in the certain domains where the curation or the specialized, actually the infrastructure is there. Like, you know, with the like for example, the COVID genome data, right? That was shared and used right away. But to be able to do that, you have the NCBI, the gene bank to preserve and share that, and people like the community knows where it is. So
I have one use case that where preserved data was reused. When I was a graduate student, somebody kept the archives of the Einstein Observatory, which was an X-ray observatory which flew for about 9 years before it ran out of coolant. And they actually made a survey that consisted of not the actual studies, but when it was moving from study to study, that's called the slew of the telescope. They saved that data because the telescope was open at that time and it was collecting data. So somebody ended up making a all-sky survey using that data of just when the telescope was moving, because it was up there for 9 years. So and doing it dozens of times per day. So that's a real interesting, saved data story.
Yeah, thanks, Jim. That's a great example, actually. Yeah, it would be interesting to see it'd be interesting to see a collection of these cases and see if that provides some guidance on best practices in preservation. We're at the top of the hour, and I want to respect people's time. So, Huajin, Susan, thank you so much for taking the time to share with us today. This has been incredibly interesting. Really appreciate you taking the time, and appreciate everybody taking the time to come and hang out with us for this presentation.
Yeah, thanks for having
us. This is awesome. Yeah, thank you.
All right, with that, does anybody have anything they want to add before we all break up? And
just one quick announcement. Next month, we are doing a multi-track call, so it's probably going to be at an unusual time. Cool. Right. Thank you, everybody.
all right, thanks a lot. Bye-bye, everybody.